More of a conversation than me just spewing out information. So if you have questions along the way or you want to make some comments, interject, feel free. You know, I won't hesitate to call on you, and I won't be insulted if you interrupt me. So with that, my name's Josh Hyman. I'm the director of the DNA sequencing facility here in the building. We'll go through this bit, and then we're going to take a little tour. I know there are a lot of you, but you get to take a tour and go in and actually see the instruments. So you'll see that, and you'll see some of the reagents we're using and flow cells and whatnot. I have a rather circuitous path that I took to get here. I started off as a mechanical engineer working on the F-15E project, so windshield and canopy systems on a fighter jet with McDonnell Douglas. I went back to school. My family was in business, and my dad wanted me to get an MBA. I got the MBA. I worked in the seafood industry for a while, and then we had a restaurant. It was Kenny Rogers Roasters. It's the wood that makes it good. So we ran that for a while. I got out of that, and then Kenny Rogers Roasters went bankrupt. It wasn't my fault, but they went bankrupt. And after that, I finally went back and did what I really wanted to do all my life, which was horticulture and genetics. So I have a degree in horticulture and genetics as well. So now I've finished. I won't go back to school anymore because my wife will divorce me if I do. But I've managed to get a few degrees along the way, and it's actually very helpful with all the technology we deal with. And as our facilities here in the building are run as businesses, it's important that I understand sort of the business end of it too, because we don't get operational support for the facilities from the university. All our salaries, fringe benefits, service contracts, reagents, the whole bit, that's all paid out of the money we take in as revenue. In fact, there's a little bit of extra money we send back to the university. They do give us support within the building because we have human resources here. We have people that handle all the financial transactions and all of that. But the core facilities don't get that sort of operational support. But we don't mind. We're happy to do what we do, and it's actually kept us a little more energized, because we have to look out to the future and always ask, what services do we need to offer? What are we going to have to close down? What are we going to have to get into? What do we need to learn? And everybody in our facility has that mindset. And I've had crew come in and say, look, we're undercharging here. I spent 4 hours doing that, and every single time it takes me 4 hours. So you can't charge whatever we're charging at the time. We need to charge about $25 more, $50 more or something, or we need to figure out a better way to do this. And so that's been good. It's kept us going, and it keeps everyone driven. Tonight I'm going to talk about the advances that have occurred over the past 5 years or so. Yeah. Oh yes, thank you. I asked you to prod me about that. Yeah, so Tom's not here tonight. And normally he would do that, the announcements. He is up in Oshkosh. He got 4 tickets for Oshkosh because he got in the air show, because he got in late last night to the air show, and they just gave him tickets for today. So he got to go last night, and he got to go today for free. So he went up there with his family. So I'll announce what's coming at you. On August 1st, we have the Archaeology of Wisconsin's Effigy Mounds. That's Amy Rosebroom, Wisconsin Historical Society. And on the 8th is the Insect Research Collection of the UW Department of Entomology, Dan Young, Entomology. August 15th is the Buzz Report, the year so far in Wisconsin insects. PJ Leash, Entomology. And finally on August 22nd is On the Springs of Wisconsin, Grace Graham and Sue Swanson, Wisconsin Geological Survey. So, if Tom asks, it was done. All right. So we're going to talk a little bit about what's going on in the DNA world. Costs are declining. They're not necessarily declining in the kits themselves, but the machines are becoming so much more productive that the cost per read or the cost per base of DNA is much less expensive. And not only that, but there are new technologies that are out there, and those new technologies do things a little bit differently than the sort of standard technologies. Illumina has been around for a long time. Illumina has certain characteristics in the way it generates sequencing. And the newer instruments have completely different characteristics, and we're using them and really enjoying what they can do for us, which has changed a lot of the services. And the instruments are also becoming more portable. Most of the companies are developing small instruments as well as large instruments, and some of them are quite small. So they can go in areas that we never could get sequencing instruments before. And finally, because of all of this, science is becoming much more personal. We're able to do things that we couldn't do before, so there will be tests where it was probably prohibitive to give those tests before. Just in case anyone isn't aware of exactly what we're talking about here and what I'm looking at with DNA sequencing, we start with the cell and a nucleus and the chromosomes inside that nucleus, but each one of those chromosomes is essentially a string, a double-stranded string of molecules that, with a ribose backbone, and those base pairs, A, C's, G's, and T's, we just use the letters for the molecules, essentially make up everything that is coding in our cells. Now, having said that, there are a lot of other things that are interacting to control that coding. There are modifications to each one of those bases that can occur, and there are interactions within the cell that we actually inherit from our parents. And as we go along, we tend to learn more and more about those things, and that's an active area of research. And we're going to compare the technologies tonight on essentially total output. What does each system do, both in the number of the DNA fragments that are generated. So we can have a system that reads many, many, many small bits of sequence, or we can have systems that read really long fragments, but fewer of them. Or we can have systems that read a lot of long fragments. So those systems, in some cases, are just coming on the market. We're going to look at the cost per base, because that is one of the things that drives our ability to provide services and make things much more comprehensive when we go in for tests. You will get to a point where your genome will be sequenced. NIH is making a big push to at least get what they say 100,000 sequenced genomes in the next 5 years, and I think they're going to push way beyond that. We're talking millions of people being sequenced. And the quality of the data has an impact on what you can do with it. And there is unique information, especially in the newer sequencers, the kind of data they generate. And we'll talk briefly about what we can do with that data. Okay, so first I'm going to start off with the total output and the cost, and give you an idea of where Illumina's at. And Illumina has a few different machines. This is the small one. This is sort of a benchtop. They have a couple that are slightly smaller than this. But to give you an idea, we'll start out and we'll say we've got 25 million reads off of a single run of one of these instruments. Now, for the longest time, we had Sanger instruments, and off of a single plate, you got 96. That's less than 100. And now we're talking about 25 million on this instrument. It generates about 5 billion bases in a single day. 5 billion bases. That's more than there is in the human chromosome, in the genome. So all the human chromosomes put together. And it costs about $317 per billion bases to generate that data. And now if you use this machine, and this is the small machine, it would cost you about $28,000 to fully sequence a human genome at about a depth of 30-fold. So that's an average depth. But essentially, 30 times the size of the human genome would cost you about $28,000. So keep that in mind as we go through. So we're starting off at $28,000 here. Yes?
So how do you like prime the DNA to go into the machine?
Well, in this particular machine, and in most of the machines, we have to create universal adapters and put them on it. So we have to have small fragments of DNA that we know, that we start with, and we usually shear the DNA and ligate, which is just a way of enzymatically joining those little fragments onto the ends. And then there's, in the case of Illumina, there's an amplification step. And that way we have all these random fragments, but on every single one of them, we know what the ends are. And then we can take a known concentration of that. We can measure the concentration and then put a known concentration onto the machine. And then it'll spread out over the machine and or over the flow cell. And in the case of this instrument, it's just a random pattern where you have a match for those little ends down on the flow cell, and we amplify a single fragment in a single spot, and we get one cluster that's all exactly the same fragment. And then we get millions of those clusters. Obviously, we got 25 million on this machine.
So when you say 30x, do you mean going over the same molecule 30 times to cover any errors?
So on average, a single base, like if you picked any base in the genome, on average, those bases are covered 30-fold deep by fragments that may be here, 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 all over, but they all line up, so there's overlapped 30 fragments at that particular spot. And quite honestly, it doesn't come out quite so nicely. The coverage tends to wobble up and down all the way across the genome.
So theoretically, with a super machine, you could 30-fold sequence a single cell.
Yes, we could. In fact, not in our facility, but in another facility, we have instruments that can take single cells and generate libraries. That's what we call them, the fragment libraries, with barcodes on each end of them. So every single cell has its own barcode, and we generate libraries of all the RNA with those, and we can sequence each one individually. We usually sequence between about 1.5 to 3,000 cells out of, say, a tumor or something like that, and we can sequence all the RNA at about a 50,000 read coverage for that particular cell. Now, if we wanted to sequence and sequence and sequence, we could go to like 30x coverage of every single gene that's being transcribed, but we generally go for a fairly low coverage on a per cell basis, but that allows us to see what's going on between all the cells, because not all the cells are the same. And you have some cells that are more progressed down one particular branch of that cancer, and other mutations that have arisen and given arise to another branch of that cancer, and you can actually pick those out and see all the mutations that are occurring together in lineages of cells.
So when somebody says deep sequencing, what are they talking about? Doing the same thing multiple times? Or?
Yes, you're covering things in a very deep fashion. So if you have cancer cells, you want fairly deep sequencing, because you want to see all the things that are occurring and see all the mutations for all the specific cells. The single cell sequencing is actually allowing us to get around some of that, because now we can actually see a single cell and see all the reads that are coming out of that cell. So that makes it a little bit easier. But deep sequencing may mean slightly different things depending on what your application is.
Sir, when they ask a question, would you repeat it? Because there are people in the back, I'm sure, didn't hear his.
Okay.
And I apologize.
That's true.
Did you say to ask questions also?
Yeah, absolutely. Okay. Absolutely. Okay, we have.
How do you harvest the cells?
How do we harvest the cells? We do some of it ourselves. So in some cases, people will give us, bacterial cultures are easy. We are now putting in a room upstairs where we're getting samples in from people. We're plating out the bacteria. We're selecting clones out of that, and we're growing them. So we're growing that up ourselves and creating a whole bunch of tubes, and then using some of those to either perform cross-linking if the cells are from an organism that has a very large genome, and using that to identify areas that are close to one another, and then we can assemble a genome. But if it's a smaller genome, we just want really high molecular weight DNA. And that's where some of the old techniques are sort of kicking in, because I would like to have DNA that we're extracting that is 80,000 bases to 200,000 bases long, because I can, as you'll see, I can now sequence that. Plant cells, if you want to harvest those, you're just getting plant tissue. And for the most part, if we're not using some of the very specific techniques we use, which is very rare, most of the time all we're doing is grinding it in buffers. So we have systems that are essentially provided by vendors now, and we look at what's in the buffers just to make sure we know what's going on, because if something goes wrong, we get to figure it out, and we don't always know how things work in a given plant or animal or whatever it happens to be. But we'll do a lot of that isolation ourselves, where you're using salts and soaps to essentially break down the cells, allow you to precipitate the DNA, wash away everything else, and you're further using an alcohol to pull off the water out of it, and then allow the DNA to precipitate, and then just spin it down and wash it. It's actually a fairly simple procedure. I don't know if you've ever seen what Tom does over here, but you know, on grandfather's or grandparents' weekend, he has the grandparents with the grandchild, and they're all extracting DNA with soap and peas and salt and everything that you can get in a normal kitchen. So it's fascinating to watch, and they're just having the best time. So, yes?
What is the difficulty of like kind of growing different cultures of like animal or plant cells or all this stuff?
Well, the animal, what difficulties do we encounter when we're trying to grow some of these things, plants, animals? Most of the stuff we don't grow. The only thing we're going to touch is bacteria and maybe some fungi upstairs. The rest of that is grown by all the researchers. So it's up to them. They have to figure out how to grow things, and in some cases we get emails asking us if we can just use the seed instead of trying to grow the plant, because a lot of times germination is a problem. So they'll just grind the seeds, and we can actually use the seeds, and depending on the seed we're looking at, we have to modify the analysis, because without getting into too much detail, plants, fertilization is a little bit different if you look at the embryo and the endosperm and look at the chromosome count in each one of those and where it's coming from. So, yes?
When you're talking about going like up to 30D, is that on the same fragment?
It's not on the same fragment. We want to avoid the same fragment, because if that has errors in that read, and along with what sort of processing errors that we've introduced, we want to statistically drown out those errors. So we want all different fragments. So taking the native fragments that we've started with, we want those to align in a random pattern. And in fact, when we see fragments that start and end at the same spot, they're usually kicked out, because we consider them to be duplicates. Even if they aren't, the risk of them being a duplicate is too great. So yes, as far as trying to get different fragments, we can do that mainly by software at the end. All right, so onward. So we're at $28,000 on the MySeq. The nice thing about this machine is it does what's called paired end 300 base pair sequencing. So this is a smaller machine, but it does have the capability that no other aluminum machine has, which is to read 300 base pairs in from each end of your fragment. Others can read in, but they don't read quite that far. So when we're doing work on bacteria and things like that, and we need certain regions or certain amplicons, certain tags out of there, and we want to read the whole thing, this gives us the ability to read up to, realistically, about 450 bases, because the quality tends to drop off of the reads as we go along. This is a machine we've been running for a while. Okay, we started with a MySeq. It had 25 million reads. This puts out about 2 billion reads. So obviously, quite a jump in output. It turns out about 167 billion bases per day. And that's in comparison to the 5 billion on the MySeq. And now the cost has come down from $317 to $52 per billion bases. And this is what we've been running for a while. Now this would drop the cost of a human genome down to about a little under $5,000. Yes?
What is the definition of a basis? I don't understand when you say 167 billion bases.
Bases. So each base is one of those molecules that is in the sequence, that is in that orientation along a fragment. So if we're reading out, and I read out ACGT, that's 4 bases. So this is reading 167 billion bases in a single day. So a human genome is about 3.2 billion bases. We usually round it down to 3 billion. So this is capable of performing sequencing on about, if we wanted 30-fold coverage, it's giving us about 2 human genomes in a single day off of a single flow cell. And I can run 2 flow cells on this, so I can get about 4 genomes in a day. But it's still, this is considered fairly expensive because this is the instrument we just got about a week and a half ago on Thursday. 10 billion reads, so 10 billion fragments sequenced all at once, all right, per day. And it generates 3 trillion bases per day. So if we do the math, we're getting about 18 times the raw output of the machine I just showed you. It's slightly more expensive to run, but because of this massive output, it's dropped the price drastically, and so now instead of it being around $50-some per billion bases, we're now down to $15 per billion bases, which means that a human genome is now, for us, about $1,300 to $1,400 to generate at a 30-fold coverage. Yes?
Do these data flows give you any anxiety to capture and to maintain and manipulate?
I tell you what, they give our IT department a lot of headaches to capturing and maintaining all this data flow, which is what the question was about. How difficult is that? This machine will fill up anywhere from 1 to 6 terabytes of storage in every single run. And I've got 2 flow cells that I can run at the same time, so every single day I could fill up to 6 terabytes. And we have storage down the basement, and we've been buying it, in 80 terabyte chunks, and it can cost about $100,000 for that. And then you look at that and you go, well, okay, that's 2 weeks. and our head IT person is having a few heart palpitations along the way, because every time we get in a meeting, he goes, wait a second, you can't do that. I go, buy it, buy the storage, I'll pay for it, and I'll charge it back to the customers, because what it comes down to in long-term storage is it costs me about $5,000 or $6,000 every time I run this machine just for storage, just to maintain enough storage to last for a while so our customers can have that available.
How much the machines cost?
This machine, just shy of $1 million. So it's not like we go out and buy one every day. Yeah. Yes?
So I do not know how these machines work, but is there one particular part of the machine's inner workings that account for the improvement in throughput? And really the way I'm thinking of this is, in the computer world, when a laptop doubles its capacity to calculate something, it's usually because of the processor and the speed by which bits travel between the memory and the processor. But the processor tends to be the key thing, that if you continue to make it more powerful, you can double your calculating power in 18 months.
Right.
Is it the same? Is it electrical processors in this thing? Is it a chemical thing? Is it a...
All right, so I'll tell you about this. What makes this more capable as time has gone on? And I'll make the analogy. You want to use computers as an analogy. It's actually easy. If we improve the materials we're using in the computer and we miniaturize them, we get a better chip, a faster chip, and we figure out a way to get rid of the heat. This is similar. They improve the surface chemistry of the flow cells that everything's going on. They improve the cameras that are taking the pictures, and they improve the reagents that are used in the process. And the big thing is getting those images in, so the speed of the system, the ability for it to extract that information is critical, because as reagents are used over a few days, their quality drops. So we have to take these pictures very, very quickly. Now the one I showed you, the instrument I showed you before this, it took about 6 days for a full run. This takes less than 2 days. So I could run 2 flow cells every 2 days on this and keep loading it. And the reason the quality stays up there is because we've got better reagents, and they have tweaked this, so instead of using, and I'll get into this in a little bit, but instead of using 4 different colors to detect 4 different bases, they're using 2 colors, so the camera only takes half as long to take images, because it's only looking at 2 channels now instead of 4 channels. So the camera's moving faster. But it's a really odd system, which I did not trust for a long time, because basically if there's one color, it's one thing. If there's the other color, it's another thing. If there's both colors at the same time, it's a third thing. And if it's a no color whatsoever, it's the 4th base. And that makes me a little squeamish. So, but that's how they've managed to do this. And they've also changed the flow cells, so they're patterned flow cells. Whereas before on that HiSeq 2500, that was random seeding of that flow cell. In this one, there are distinct wells, and the way it's set up is there's a layer on top of the flow cell outside of the wells that doesn't allow anything to attach to it. And then down in the wells, that's where you have all of our fragments that will anneal and extend our DNA. So that allows us to look at certain points and know that it's all going to be clustered in a certain fashion. And that also speeds up the camera, because it knows that it has to sit and have a specific layout of the cell rather than just random spots where it has to figure out where every single cluster is. Yes?
So just going back to your lab, what do you think the percentage of human or animal DNA that you do versus some sort of plant DNA? Like, what do you typically sequence for?
So the question is what is the breakdown of the types of samples we get, plant versus animal versus bacterial? We usually don't look at it that way. We usually look at it as what's coming in as far as the type of services, RNA versus DNA versus, and RNA is always been a fairly large chunk of things, and that a lot of times we don't even know what organisms are coming in, because we do allow our customers to prepare their own libraries, all right? And they just hand them to us and say, sequence this, please, and we sequence it. So that we don't know, but I will tell you that we, about a little over a year ago, we signed a license with KeyGene, which is a company that has a process to generate tags, DNA tags throughout a genome. And depending on mutations in the genome, you'll get different tags. All right? So if 2 plants have, you're crossing 2 parents and then they're segregating back out, and some of those markers are dropping out, and in other words, the specific genes are slightly different, mutations are slightly different, you'll be able to detect that. And we signed a license with them. We have probably 1/3 of what we're processing right now is just those types of libraries, and they're mainly plants. but people are using them for fish populations, for ground squirrel populations, for insect populations, for bird populations now. They're using them for everything. Another, pardon?
Do you do any clinical samples?
We do some clinical samples, and we would like to do more. We have to get participation by the west end of campus, and Stephen Mine is down there now, and he's heading up the Human Genome Initiative on the west end of campus, and we've had a lot of conversations with him. He's excited about what's going on here because we have a clinical license. We are certified, CAP-CLIA, registered and certified, under the Wisconsin State Lab of Hygiene's umbrella. So they're the ones that are responsible for our registration and certificates. And we want to do more of them because we're not getting that many right now. And there's a fairly large investment in getting everything. I mean, you have to jump through a lot of hoops, and you have to have a lot of safeguards in place and so forth. Yes?
How do you like keep the DNA from being contaminated
With other DNA?
Let's say like from bacteria floating in the air or a sneeze that somebody has?
That's a very good question. So how do we keep one sample from being contaminated with another sample? A lot of that is the flow within our facility. I'm going to show you the sequencing room. What I'm not going to, with all the sequencers in them. What I'm not going to show you is what we call the pre-PCR room, where we're taking raw DNA, which is usually highly concentrated from one sample, and preparing libraries. And I'm not going to show you the post-PCR room, where we've done amplification, and now everything has that universal tag on the end of it, those adapters. And those rooms are cleaned every single week. We essentially take Clorox and wipe down everything, including all of our pipettes and everything else that we're using to measure things. All of our reagents come in and they're used for a week, except for things like polymerase, which are, obviously we're not going to throw those out. A polymerase is an expensive enzyme, so you don't throw tubes of it out. But the water gets changed every week. Standard solutions that we can mix up, which are fairly inexpensive, get thrown out every week, and we start with a fresh batch. We go to the chemicals cabinet, we measure it out, it's sterilized, it's run through our system, you know, we have filtration or whatever, and then we start using it, and it's only used for a week. And part of that was CAP-CLIA, part of it we were doing already, but we've really locked that down. Our lab is very clean now, especially compared to some of the labs I've worked with in the past, where we were dealing with a lot of plants, and so we'd bring in plants and all the soil and everything came with them. But yeah, we keep things very clean, and we have to be very organized. We have thousands of samples that go through, especially with the genotyping by sequencing under Keygene's license. We get multiple plates. We can get a few plates from somebody, and each one of those plates has 96 wells on it, so there's 96 samples. We have about 14 plates of samples right now that have to be DNA extracted from them. And we have 400 plates of different DNA samples that we just sheared this morning. So we get large batches, and we have to be very careful. And the worst thing, the worst feeling in the world actually is to go, okay, did we get these two samples switched? Because it isn't just cross-contamination, it's actually switching samples that we have to worry about. So we have some electronic systems where we can barcode the outsides of tubes, just like in a grocery store, you go and you swipe and you know exactly what you have. Yes, in the back.
What can one learn from DNA sequencing? What's the purpose of it?
What can one learn from DNA sequencing? We can watch disease spread. We can see how disease spreads from location to location and see how it's spread by comparing cultural norms, burials, handling the dead, handling sick patients. We can see what's happening in hospitals. NIH has a clinic in Frederick, Maryland, where they got a very resistant pneumonia infection spreading throughout. And they had to use DNA sequencing and look at all of the mutations in that particular strain. And in this case, I think it was bacterial, and it was spreading throughout, and they could look at all of the mutations in it and identify which patient had a particular strain, what mutations occurred, because they were swabbing mouths, they were swabbing armpits, they were swabbing the patients, and they could actually see it mutating as it traveled through the patient. And then they looked at sinks, they looked at bedding, they looked at gloves. They could actually identify people in that clinic who had bad hand washing habits and find out who was spreading it, and it allowed them to clamp down on it. It's been used extensively in areas where we have epidemics, Ebola, Zika virus. Believe me, there was a lot of sequencing that fast on the Zika virus. HIV, we look at everything like that. We can look at plant populations and see what's phenotypically yielding better results, whether it's the oil in the plant or growth rates or whatever, disease resistance. We can look at that. And we can identify all of the changes on the genetic level that are giving rise to the downstream effects. We can look at populations in lakes without even seeing the fish that are in there. They're testing the water and looking for DNA from Asian carp. And so they're trying to detect where it's gone, what rivers it's in, just by this DNA it's shedding in its feces, in its skin, or scales, in its whatever. So we don't do any crime scene. We don't do any CSI. But what we have done, which was interesting, is I'm trying to figure out how long ago. So this was 2014. So we did some crime scene type work, which was not really crime scene, but it was identification of a World War II soldier who was killed in France. And then no, this was before that. This is what got us involved in that. So this is PFC Gordon. Lawrence Gordon, his namesake, was living in, I want to say Alberta, but he's right on the border between Alberta and Saskatchewan, so I don't know exactly where he lives because he goes back and forth over there. And at any rate, so his uncle, obviously, was killed in World War II and was buried somewhere, and they didn't know where. So there was a trail, a paper trail, that was followed, and as it turns out, it looked like he was buried in an unmarked German grave in France. So there are German cemeteries in France. Interesting note, German cemeteries in France are leased from the French government. All other cemeteries in France are owned by the country whose cemetery it is. So the American cemeteries are American soil. The German cemeteries in France are French soil. So obvious reasons there. But we managed to work with the French authorities, with the German authorities, and with folks here, and we got a positive identification. Another lab in the United States got a positive identification. We brought the remains home. Yes?
I'm guessing that there's not like this little thing where he pricked his finger on a needle and then you had his DNA and it's like, oh, 99% match.
Teeth. Teeth and bones. Teeth are really good because teeth are essentially a closed vessel, a very good vessel. It's almost like having a glass, a sealed glass, and you can drill down into it. And some bones, if bones have been exposed to heat too much, and he was in an armored personnel carrier which burst into flames, so there was that problem. So we had some bone tissue, and we encouraged them to take teeth, back teeth out of the upper jaw, the upper mandible. And so we had that, and we had the bones, and that powder was distributed because it was ground. So it was distributed to the French authorities first because it was on French soil. So they had to do things first, and they were anxious to do it because they sort of wanted to show up the Americans, because the Americans wouldn't participate. Our Department of Defense wasn't as interested as the French were. So they did it first, but it was all with bones or with teeth. We didn't have anything else. We didn't have hair. He actually was wrapped in a German overcoat when he was buried the first time, and that was part of the problem. Even though he was right next to the soldier that was killed at the same time in the same armored personnel carrier, they should have known from that, but at any rate. Yes?
What process would lead the mandible to the identification?
It's just the teeth themselves. So they pulled a tooth. So we pretty much knew it was him. So that's the only reason we would look in that area, is simply to have preserved DNA. Yes?
When you're getting these machines that can do more and more, so much more,
Mm-hmm.
do you need more personnel to prepare the samples?
So do we need more people to prepare the samples? We lost one person about a month and a half ago, and we've hired 2. So that should answer your question. We are providing so many more services. We're expanding what we're doing right now, and it's becoming more complex. And in some cases, people will say, well, why are you doing this? We can just go out and get our sequencing at X, Y, and Z, or wherever it happens to be. That's not our job, really. Our job is to provide the information and the services. Not the sequencing part is actually easy. It's everything prior to the sequencing where we really have somebody there that has hands-on experience, that has failed a lot. Because by the time we get a service out the door, we have worked and worked at it and banged our heads against the wall. And so our people that we've hired really know what all the pitfalls are. And so yes, we need more people. And so we're hiring 2 more people, both of whom have sequencing experience and have prepared libraries and all of that. And one of them worked on Asian carp. And then so I actually got to see this little story about what she was doing just recently. So. All right. Now, the question is, what do you need a sequencer? What do you do when you need a sequencer in the field? Well, we've got this big NovaSeq, and we obviously can't take it out in the field, but there is a smaller sequencer right here. Can you see it? You can see that all right? Okay, so how about this? Can you see it? It's that simple to take a sequencer with you out to the field. Sequencers like this recently went down to Antarctic, so I'll skip forward on a few of these things, but they went down to Antarctic. They've gone to the pink lakes in Australia, which are really cool looking, but have different microbes growing in them that give them that pink look. This weighs 87 grams, so about 0.2 of a pound. You can carry it in your suitcase. You hook it up to a laptop. The problem is analyzing the data. So you collect data, but you have trouble analyzing it because, surprisingly, I can run this and take more space from a run on this than, say, a single lane on a HiSeq 2500. This can take up to a terabyte of space by the time we start analyzing it. And I'll show you why in just a little bit here.
But you still have to prepare the DNA for it. To what extent you can prepare the same?
Yes, you do have to prepare the DNA, but you can take those reagents and get a decent preparation in some of these locations. So yes, the question was, do you still need to prepare it? And right now you do. This company that produces this is working on a little system that will prepare this most of the way for many samples you get, but not all samples. you get.
So before you were talking about splicing your DNA before running it through. Let's say that you have that little pocket-sized sequencer and you're in the field analyzing a tomato plant for some reason. What steps do you have to take before it can go into that?
Well, right now, we isolate the DNA, and we go through it. So I can't run this in the field. I don't have a field lab that I can just take my reagents and take everything I need. We would extract the DNA. We would then run all our enzymatic operations on it. So, I mean, it takes a good 2 or 3 hours once we have the DNA extracted to generate everything needed to go on one of these instruments. But they have taken them down. I worked with a group that went down to the Antarctic. So it's not a simple thing to just run out into the field with us and go, hey, I'll just take a leaf and jam it in here. You don't do that. It takes a little bit of work. The nice thing about this is it's not subject to vibrations and damage that way that one of these larger instruments is. So once we get one of these larger instruments in, we have to calibrate them and they have to sit there. If we move them, they have to be recalibrated. This I take in my pocket. I take the flow cell, stick it on here, load my library onto it, and run it. So, all right. So the nice thing about this particular technology is I can get really long reads. And this is where going back and getting very, very high molecular weight DNA that's in very long strands, 80 to 200,000 bases long, is important because I can sequence contiguous strands and it'll run them all the way through. The longest fragment that's been sequenced on this particular platform was over 1 million bases long. One long strand of DNA all the way through. In fact, there are people on campus that have done that. We aren't shooting for that. We don't want the longest read. It's great to be able to say, hey, I've got the longest read on this instrument. What we want is that range, because that range, 80 to 200,000 bases, actually allows us to assemble whole genomes from smaller organisms very, very quickly. They're draft assemblies because they have a lot of insertion and deletion errors in them, so bases, it won't count a base, it'll just jump over it. It'll think it has 3 when it actually has 4 bases in that location, with like CCCC, which I'll get into. But that's the downside to this, but the upside is it gets all the structure right. It has some small errors, but the big structure that we're seeing is correct. And because it consistently messes up, we're able to take all the fragments from this and put them all together, and they assemble nicely, and we can sometimes assemble whole genomes in a matter of 15 minutes or so off of these reads, which is phenomenal when you consider how long it used to take us to clone all these different fragments. Yes?
If you had to look back in time at the best machine, full-size lab machine of each year, what is that comparable to in terms of power?
In terms of power, how do you mean that?
Capability over time. That handheld unit, how many years in the past would you have to look to find a full-size lab machine?
That could do what this does? You actually couldn't, because we never could. The longest read we could get on our Sanger instruments, and we still have one of those, but they were the workhorses for a long time, was about 700 or 800 bases long. That first little machine I showed you, the Illumina MySeq, can go out 600 total bases. And before that, there was an instrument that was trying to hit between 700 and 1,000 bases and do a lot of random fragments. It's no longer available, but that was the limit of what it could do. And so this is so much different. It's not the total output that counts in this case. It's the length of the read, and we've never had that capability before. This has opened up a completely new area for sequencing, because we can now see the structure and get it correct, because there are a lot of repeats in the genome. So you go through one fragment, and then you see the exact same fragment right next to it, or sometimes flipped, or sometimes 2/3 of that fragment and another 2/3 of that fragment. And the short reads that are coming off the Illumina systems cannot handle, you can't assemble there because it's all the same and it keeps repeating itself. So you don't know what you have there until you have this very nice contiguous fragment that starts with something random in the beginning, that ends with something random in the end, and then has these repeat units in the middle, and you can actually count all the repeat units, which means that those diseases that are dependent, like Fragile X, that are dependent on having a lot of repeat units all in a row, can now be read with this and with a packed biosystem, because we have these nice chunks that are very long and can start before and end after all of those repeats are lined up. So we can actually say, yes, you have 312 CGC repeats, and I'm afraid that that's not good news. You know, yes, there's Fragile X, that sort of thing. So completely different mindset now with this. Yes?
Another basic question. What's physically inside that portable unit that physically can do the work? Is it a solid state thing, or is it
I will show you.
Okay.
Okay. All right, so this one, about 10 billion bases per day, and we're looking at $90 per billion bases. So a lot more expensive than the NovaSeq, but on the other hand, completely different idea here. So we're not trying to compete. I mean, the companies will say they're trying to displace this other company and so forth. But from our side, our service and application side, we don't think they're competing. And I wish they wouldn't be in that mindset. I wish they'd switch out of that, because then they come in and act like, oh, ours is so much better. And we're going, no, it's just different, you know. So at any rate, it is more expensive. It would cost you about $8,000 for 30x coverage if we had really good runs every time. So that 10 billion bases can sometimes be, I hate to say it, less than a billion, depending on the quality of the DNA and our library preps. So it's a bit more fickle, but it's especially sensitive to DNA quality. And then we get into essentially what's 10 times this, and we have one of these on order. We're trying to get it in, and it can generate essentially what 5 of these do, so 50 billion bases per day, same price range. And then finally, the same instrument line. What you can't see very well is there are 48 small flow cells, which are actually much denser than the flow cell that goes on this. So it's getting close to the output of the NovaSeq, 2.4 trillion bases per day.
How big is that?
That sits on your desktop. It's about that wide and about that deep. I think I checked the measurements on it, and it is, they had it in millimeters, 440 millimeters.
And
So
how big is the NovaSeq?
The NovaSeq, you'll see, stands about this tall, about that wide. So this was sitting on it, and it was sitting right about there.
So it's like a photocopier versus a whatever.
Yeah. Oh yeah, huge difference.
How do you cut the DNA so that?
How do we cut the DNA and so we can put the fragments on the end? We can either sonicate it with sound waves, and that'll blast it apart, and we control how much sound hits it. But what we'd rather do is cut it with enzymes. So there are enzymes that'll create nicks or cuts in DNA, and if you give them a little bit of time in the right buffer, they'll generate the amount you want. If you give them less time, we get slightly larger fragments. So that's how we cut DNA.
And do you ever use technologies like gel electrophoresis to make sure that you have the right length?
We always do. We always use gel electrophoresis to find out the size, because we need to know what size fragments, especially on the Illumina system and then on the packed biosystem, are needed to do that. So this system is getting down more towards the HiSeq 2500, which we've been running for a while as far as cost goes per billion bases. So we could run a human genome through this. And somebody sent, and they actually didn't use this instrument, they used a lot of these and published earlier this year. And you really saw the power of this. They found regions we haven't been able to sequence before. Many, many regions we hadn't been able to sequence for or get all the way through and understand how that region was structured in the human genome. And they also identified and could locate telomeres. So telomeres are at the end of chromosomes, and basically you have a string of adenine residues. You just have one residue on the end of it. Did I get, I always, yes. I have to remember if it's A's or T's on the end now. At any rate, they're long, and it protects the chromosome. And the length of that is indicative of the life that's left in cells. So shorter telomeres, as you get over, your telomeres shorten, and you start seeing more cellular damage to the DNA because of that. And they were able to size and locate 17 out of our 23 chromosomes, so we would have had 46 telomeres. Actually, since we're diploid, we would have had 92. But since we're only looking at, one copy of the genome at that point, or a haploid copy, so just not both sets, they would have had 46 that they would have to dealt with, and they located 17. 17. That's an almost impossible task even to determine the length of one of those, and they sequenced 17 of them and positively located them. So that's the power of this, and going forward I think you're going to see that more and more, especially with this instrument, which I'd like, but we just don't have the extra $1.3 million to get the price down to where it needs to be. Yes?
Is there a way to not mix up the bases because the strands of DNA are described in pairs of bases?
We only sequence one side of that at a time. So is there any way to not mix up bases? Well, we're only sequencing, this is double-stranded DNA. It's always denatured, and we always sequence one side of it as we go along. So we're only getting one copy, not one copy, but essentially one look at one strand, and we infer the other strand because A's always pair up with T's, G's always pair up with C's. So we know what one strand is, we know what the other strand is. What we don't know is any modifications to the DNA. We can see them on the one strand, but if there are modifications, there are, without getting into details, there are modifications that can occur to every single one of those molecules, and it affects expression levels of that. So we may not get RNA out of it. All right, this is coming. It hasn't been released yet. This is a smidge ion. You can actually have a sequencer that's attached to your phone. Take it anywhere. I'm not sure how much information you'll get. I'm not sure exactly how you'll analyze that information, but this will be available for you. Anybody can do this. I'm not saying that anybody can analyze it, so you may have a lot of information that you can't do a thing with. But you can at least generate information. And we could see this happening if you had very specific assays and you were in a remote clinic, so you weren't near a facility that could run the sequencers. You could run something like this, and if you had specific assays, you could go through and just run those assays so you know that you're targeting certain molecules and you're only going to be running those molecules. You could run through them, get sequence information off of them, and immediately have an answer. Now that also assumes that the people in the clinic know what that information means. So even if I find mutations, there are so many different mutations in the human genome that have effects on disease or growth or whatever else. And combination of those may offset one another. And doctors deal with a phenomenal amount of information. And with 3.2 billion bases in the human genome, they might be a little overwhelmed with a lot of it. So that's why we have people that are simply there to analyze what is available in that information. And there are thresholds for every single mutation as to whether or not it's going to be discussed. And it'll have a true impact on somebody's health.
Well, yeah, there must be a big error rate, a variation of errors when they come to interpreting that.
Yes, there is a big error rate in interpretation. There's also a certain error rate in sequencing. So we may perform the sequencing and then we give some indication to a clinician that there may be an issue, and then we turn around and we resequence it in some other fashion to verify that our first sequence was correct. And then the error rate in interpretation is huge right now, and is always, for every kind of mutation, it tends to change over time until we get a solid idea of what that mutation will do and whether it will do it in all populations. So we are very similar to a lot of other organisms, very similar to swine. Physiologically, we have swine here that we've made mutations in to see what will happen if we make mutations in certain genes. We're studying, my boss here, the associate director of the center itself, is working very hard on neurofibromatosis, which is cancer of the nerve sheath. We have swine that we have created single and double mutations in the NF1 gene, which is the gene responsible for that disorder. We've also put a mutation in the P53 gene, and the P53 gene, I don't know how they get these names, but at any rate, I'm sure there's, like if I looked it up, there'd be a reason somebody named it P53. But the P53 gene is responsible for DNA repair mechanisms and essentially helping avoid cancer. It's a big gene when it comes to oncology, all right, because you want to repair things and you also want to identify cells that have cancer, and it's involved in that whole pathway. We made double mutations in that, and that particular animal there was so much cancer in that was springing up everywhere in that animal's body that they had to put it down. It was great from a research side to sort of understand what was going on with these pathways, but everyone was a little horrified by it. You know, you don't want to cause another animal pain, and so very early on they saw what was going on and put the animal down. But we're very capable of picking off certain genes and finding that they have a large effect. And we can prove that they have a large effect because we do have the ability to mutate single bases. We can excise them and create deletions. We can insert bases. We can insert large chunks of DNA now, very specifically. And we can definitely change one base to another base. And that's what was done in this case. They just switched them, altered it, and this is a very personal quest for a lot of us, because we know of children that have NF. And in fact, Chuck's family has been interviewed. He's been in the newspaper and on a few shows because his son has NF. So they've interviewed him, interviewed his son. So it's a very personal journey for a lot of us.
What's NF?
Pardon? Neurofibromatosis.
Oh, okay.
It's the cancer of the nerve sheath, which means it's throughout the body, but a lot of times it manifests itself. You'll see elephant man, nerve sheath tumors. So,
do they have a standardized software that assists a clinician with analyzing the data, or is it just everybody seems to have their own particular perspective and proprietary software?
There are a number of software packages that people tend to use, but then again, there are a number of packages that allow you to take the whole genome and look at all of the mutations, and then it just pulls out those areas where we know the mutations are causing problems, and it looks at mutations that we know will cause a difference in expression of the protein, the structure of the protein, because every 3 bases is an amino acid. Essentially, we're dealing with 3-letter words throughout the genome. And we know for the most part, where these are starting and stopping. So if something shifts a little bit, we get a deletion. It shifts everything and changes everything downstream from what's being expressed. And so we can see these, and the software picks that up and says, this is a same-sense mutation. It shouldn't have any effect. All right, because those 3 letters, that last letter doesn't always affect what amino acid is being, yeah. What, pardon?
What that last
Yeah,
letter,
because there's wobble.
that combination
Yeah,
is coding for.
is coding for it, yes. Thank you. So yes, there is different software, but for the most part, that's what the software packages do. They tell us when there are these same-sense mutations, missense mutations, or nonsense stop mutations.
So there is kind of a standardization within the
There's
software.
standardization, and there are groups that are looking at mutations and deciding which ones are critical. So there is a large body that is watching this all the time. All right. So finally, we have the PacBio sequel. This has been around for about 5 years. This produces far fewer reads. It has a smart cell, basically with about 1 million wells in it, tiny wells. But we can generate up to about a 70,000 base pair read with this, and about 25 billion bases per day can be run off of it, not because that's how much a single smart cell produces, but we can run about 2.5 smart cells a day. All right, they take about a 10-hour run, and we immediately start with the next smart cell and the next smart cell, just one after another. It does cost a fair amount. I put down $120 per billion bases. I put 90 for the Oxford Nanopore, but the truth of the matter is we throw out a lot of data from the Oxford Nanopore because we just can't use it. It's too low quality. So it's slightly more expensive on the Oxford Nanopore than I had listed, but at any rate, this gives you a good idea of what this can generate. It would cost you about $11,000 for 30x coverage of a human genome. Given the output we've had recently, we just jumped up to about twice the amount of data we were getting 3 months ago. They update chemistry, they update software, and all of a sudden we're a whole lot better off. We really like this machine because what it can do is loop around and around and around and read the forward strand and the reverse strand. So it keeps them together, and you'll see how it does that.
So which stock did you buy?
Which stock would I buy? That one is overpriced. I mean, they don't have that much of the market.
So which of the 3 would you buy?
Which would I buy? Illumina's been a very good one. Watch these guys. They're very hungry. They may be overpriced right now. I don't really look at.
Who do you think's coming?
These Oxford Nanopore is definitely coming on strong. I think PacBio will stay very stable because they generate data that's longer and it's higher quality, much higher quality, because as you'll see, I can read back and forth over the same fragment, the forward strand and the reverse strand. So those, a double-stranded fragment, I can keep reading both of them and get rid of all the errors that are being generated in a single read. And so I can go out about 8,000 bases and get this beautiful chunk of DNA and use that to polish up genomes and know that I have a long contiguous piece that's good. Whereas this, I get a very long contiguous piece. Yeah, exactly.
But Illumina dominates the market right
Oh, Illumina is just, it's, yeah. Illumina is huge. So as far as sequencing goes, they're very different. As I mentioned, what we do with Illumina is we take fragments and we amplify them. And so when we sequence, we're actually sequencing a cluster of fragments at a time, and they've gone through some amplification. So any of those modifications that we saw on the native DNA are gone. They don't exist anymore. And they're short fragments. Oxford nanopore, really, really long fragments, but it's a single pass through a pore. That's it. All right, you got one chance to catch it. PacBio, not quite so long as Oxford nanopore, but you get to sequence it again and again off of the same strand. And so you get many, many reads off of a single strand. And so you can use that information to detect nucleic acid modifications, methylation, things like that, which may not mean anything to you, but it does influence what gets expressed in a cell. All right, to give you an idea of how they read things, if you look at each one of these dots, they're monoclonal clusters. They just show up, and that makes it look nice and pretty, but the truth of the matter is when you look at the image itself, they are dense. They are almost one on top of another, and the software has to be able to discriminate every single base in there. So they use some fairly sophisticated software to figure out which pixels are showing up consistently as one color and changing every single cycle and still staying uniform within that little area on every single cycle. And so, yeah, just give me just a second. So what happens is we have strands coming up, and we have a little primer. It's a little chunk of DNA that we can anneal to the template that we have. So this is our template. That bluish gray area is primer. And we add a base. And the base that we add has a little blocker on the molecule. So we can't add more than one base. And that base also has a fluorescent tag. So we add it in one cycle. And then once we've added it, we take a picture of the flow cell. And when you see the flow cells, you'll go, how do you get 250 million fragments in one little area? But they managed to do it, and so the cameras are phenomenal. You remove that blocker in another cycle, and then you start again. You add one more, you wash everything away, and then you add one more base, and you take a picture. So we know the cycle by cycle, if I perform 100 cycles, I get 100 bases added to it, and we know exactly how long that's going to be.
So on that slide, the squiggly line is the DNA that's being sequenced, is that right?
This is our template, and that T is our signal. So we're actually getting the reverse complement of our template up there, because we've labeled this T, and so we know what's it. And this squiggly line is light.
The green stream of beads is what's being seen.
Yeah, this is our template strand. This is where we've attached to the flow cell, and this is the one that's giving our signal. So if we read this, we're going to read T, G, C, T, A, C as we come down, and we're going to add them one base at a time. So that's how that one works.
What's happening is you're putting all the 4 bases in there and see which
ones stick to which cluster. Yep, we can flow them all at one time. So all 4 bases get added. They stick to different clusters. They tag them with a fluorescent tag. We read it. We take a picture of it and look at the frequency, the wavelengths coming off, rather. And that goes into the software, and away it goes. And then we strip off the blockers, wash everything away, and we add another 4 nucleotides that are all fluorescently labeled in the next round, and do the same thing over and over again.
So the thing that's different for each base is the frequency of light that's absorbed as a result of.
That's correct. So the differences between the bases are simply the fluorescent tags that get added, and then once the blocker is stripped off, the fluorescent tag is also stripped off with it, and so therefore it no longer puts out any light. Yes?
The DNA won't just perfectly line its bases up. How does it distinguish between randomly placed dots?
Okay, so the question is, how does the DNA do this? There are bonds that have to occur between bases, and they only occur in one particular conformation. And so you don't get a T joining up with a C. A T will always join up with an A, and a G will always join up with a C. So whatever we have on our template, we'll get the reverse, what we call the reverse complement of it over here. So if you look at this, A's and T's are always together, G's and C's. And if I have a C over here, I'm going to have a G over there. And if I have a G in the first place, I'll have a C over there. So that's how it will always work. We're pretty sure of that. So it's worked for a long time, and if it stops working, we're in trouble. All right, so Oxford nanopore sequencing is completely different. You have a membrane that essentially is impervious to any ions. And then you have a voltage difference across that membrane, and you have protein pores that are designed so they will change conformation and change conductivity based on what's going through that pore. And what happens is we essentially attach a different protein to that strand. That's our adapters that are added to our fragments. And that's all we do. We just add that to it. And we put it on this system, get it nice and clean, put it on the system. And it essentially unwinds the DNA. It takes the double-stranded DNA and unwinds it. And takes one strand, the leading strand, will go down through the pore because DNA has a charge on it. So it will migrate through the pore. And as it migrates through the pore, the motor protein that's attached to all of this, and I don't know anything about the motor protein, but it works to keep everything going at a certain speed. That pore can detect about 6 bases, 6 different molecules at a time. So it basically gives a signal for 6 molecules, ratchets down 1, and takes a signal off the next 6. I think it's 6. It's 5 or 6, and they've changed over time. So, but it gets a different signal every single time, and the signal that comes through is recorded. So what is the current at this point, at this point, at this point, at this point? So as it's ratcheting through, it's picking up that signal. And depending on the strength of that signal, it decides whether it's a G or a T or an A or C's. And as you can see, it's pretty amazing that they've managed to do this, but you could have some error in here because you've got one shot to do this, one chance, and it's a molecule. I mean, it's tiny. I mean, this is about as small as you can get as far as we're concerned. And as it runs through there, it's not running at a base a second or 10 bases a second. Right now, I think they have it at 75 bases a second. So it is really streaming through there quickly, and they're talking about ratcheting up to hundreds of bases per second. So when they push it up to hundreds of bases per second, all of a sudden, the output of this system will go up. So right now, that 2.4 trillion bases per day, if they double that or triple that, we could have, 7 to 10 trillion bases per day.
Is it a single nanopore or is it an array of nanopores?
This is just showing one, but there's a whole array. There's 452, I think, on the flow cell that goes on this. There's about 6 times that on the Prometheon, on the big machine.
So presumably you could find, so you could get the sequence off of each nanopore.
Oh, yes, you can. They aren't always occupied. So you can get a sequence off of every single nanopore simultaneously, but they generally aren't occupied all at the same time. And because they're so small, if there's anything in there, they get blocked very easily. They can get clogged, and they do. So it starts off giving you a tremendous amount of information at first, and then you watch that taper down and taper down and taper down. And they boost the voltage periodically to maintain throughput on those things and I guess try and blow through some of the debris that's in there.
But you would end up with multiple readings, one from each nanopore that you would compare and see where, throw out the ones that were erroneous and keep the ones, you know, if there were 5 sequences that had one base at a position.
Correct. So the question really revolves around how do you make sure that a strand is correct, that the read you're getting is correct. And the only way you do that is you compare across strands. So you have all these different strands that are going through, and you take everything that's overlapping. You know, we talked about coverage. So what you look for is individual errors. So if I have something that's a diploid, an organism that's a diploid, and we should have 2 copies, and they're exactly the same, all of my DNA should be exactly the same. If it's diploid and I have a mutation in one of the copies of the gene and it goes through, I should see 50-50 roughly, right? 50% of the copies with the mutation, 50% without. So it's a statistical measure that we use to determine whether or not a difference in that sequence is a true difference or if it's an error in the sequencing itself. And so that's how this system works. And because we're using native DNA, we don't have to do any amplification, and we're just sending it through the pores. As long as this thing's running, it can sequence. And as long as it isn't clogged, it'll just continue to sequence and sequence and sequence. In reality, we hit about 24 hours, and we're barely getting anything through the pores. They tend to clog, they tend to break down. It's like a battery, it can burn out or lose its charge, and that's what goes on with this system. I mean, we only have so many ions that are responsible for the charge difference across these pores.
So how do you reset it?
How do we reset it?
Yeah, if it gets clogged.
We use it up for that day. We take that flow cell, we throw it aside, and put a new one on. Yeah, that's the only way to reset it. They do have a reverse where they reverse polarity briefly, and I'm not sure how they're doing it. They seem to be figured out a way to do something with each individual pore to do that, which to me is a little bit of magic, but you know, it's their magic and it seems to be working for us, so I'm all for it.
Are they glass?
These membranes?
No, the pores.
The pores? No, they're protein.
Oh, there. The array is the protein?
So I'm not sure exactly what makes up this membrane.
Oh.
And they're not about to tell me. So you know, they give you a rough idea of what everything is, but you know, that's a billion-dollar question that they're not going to give you the answer to. And finally, we have the Pacific Biosciences Smart Sequencing. And essentially what we do on this one is we take fragments, and we generally like to fragment down to the 20 to 25 kb range if we have one sample going on. If we have multiple samples going on, we have to bring them down to about 8,000 base pairs, because we have to be able to read barcodes that'll be on the ends of the fragments. And if our fragment is too long, we may only get one pass of the barcode and we won't get a good read on the barcode, and then we don't know which sample it is. So we like to run these at about 8,000 bases, but the nice thing about these constructs is that our adapters are circular. So when we denature these to get a single strand, it opens up into a circle. So when I sequence, I take this, I open it up into a circle, and I don't know why I had the wiggle here, but essentially you're going around and around this, so you get a forward strand, a reverse strand, forward strand, reverse strand, and what we'd like to do is cover that at least 5 times. And if we do that, the accuracy is phenomenal. Don't tell Illumina, but I trust the accuracy of 8 kb and smaller fragments more than I trust the accuracy of what we're getting from Illumina. And the other nice thing about both this and the Oxford Nanopore system is because there's no amplification, we tend not to have what's called GC bias. So fragments that are either GC rich, and GC bonds are very strong relative to AT bonds. So fragments that are GC rich tend to be difficult to amplify because the bonds are so strong, and AT rich fragments tend to be difficult to amplify because they're so weak they tend to fall apart on you. So what you see with Illumina is below about 20% GC, you don't get much sequence. Above about 75 to 80% GC content, you start losing your coverage too, because you just don't get those fragments amplifying and sequencing. With these systems, we don't do any amplification. Whatever we have in our pot to begin with is what we put on the machine, is what gets sequenced. So, and if you want, I can show you this little YouTube thing that describes how it gets sequenced, and I'll try and only show you about a minute of it, if that's all right with you, because the rest of it gets a little wearing. So let's just see if it works. So, yeah, it is getting late. I mean, I can wrap up in almost no time. We can get over and take a quick tour if you want. Okay, we'll take the YouTube. 1 minute. We'll see if it works. And I'll immediately move to about there, I think.
Duplicate entire genomes in minutes by reading the DNA and sequentially building a complementary strand with matching building blocks called nucleotides. Pacific Biosciences smart sequencing harnesses the power of the polymerase as a sequencing engine by eavesdropping on it while it works to replicate DNA. This approach is enabled by 2 proprietary technologies. The first is phospholinked nucleotides. To visualize polymerase activity, a different colored fluorescent label is attached to each of the 4 nucleotides, A, C, G, and T. In contrast to other sequencing approaches, our phospholinked nucleotides carry their fluorescent label on the terminal phosphate rather than the base. Through this innovation, the enzyme cleaves away the fluorescent label as part of the incorporation process, leaving behind a completely natural strand of DNA. This enables us to exploit the inherent properties of the DNA polymerase, including high speed, long read length, and high fidelity. The second key technology is a nanophotonic visualization chamber called the Zero Mode Waveguide, or ZMW. It enables observation of individual molecules against the required background of labeled nucleotides while maintaining high signal to noise. The ZMW is a cylindrical metallic chamber approximately 70 nanometers wide that is illuminated through its glass support, creating an extremely small detection volume, just 20 zeptoliters. Nucleotides diffuse in and out of the ZMW in microseconds. When the polymerase encounters the correct nucleotide, it takes several milliseconds to incorporate it, during which time its fluorescent label is excited, emitting light that's captured by a sensitive detector. After incorporation, the label is clipped off and diffuses away. The whole process repeats, creating sequential bursts of light corresponding to the different nucleotides. These are recorded, thus building the DNA sequence. Our smart sequencing harnesses the polymerase's natural ability to synthesize 10 or more bases per second over thousands of continuous incorporations, leading to high speed and long read sequencing. Additionally, our smart technology design allows simultaneous multiplexing of thousands of ZMWs in parallel, all concurrently replicating DNA in real time. The data is assembled into whole genomes and analyzed by researchers. Our smart sequencing technology will ultimately translate nature's ability to replicate an entire genome in under an hour to an instrument that will sequence a human genome in minutes for under $100.
All right, so I hope you followed that well enough. Basically, they have really tiny wells, and the polymerases that are bound to this also get bound to the bottom of the well. And so they're stuck down there. And because of the shape of the well, not much light gets out, only the light that's at the very, very bottom of the well. So as they incorporate nucleotides that have that fluorescent tag attached to them, they hold on to them for just a little bit, and that's the wavelength of the light that comes out. Everything else is background noise, and it's fuzzy, but all of a sudden you get this one peak that comes up. And so that's how they sequence everything. And it can just keep going around and around and around that circle as it's down in the bottom there. And the shorter fragments do a little bit better job because it's just easier for them to get through the system. And that's how PacBio sequencing works. Okay. We've talked about most of this, the quality of the data, the unique information that it has. Error rates are very high for Oxford Nanopore, 1 in 5 to 1 in 15. We can see the error rates on them. We get quality metrics off of it. We only use the better quality stuff. Pacific Biosciences, we get quality scores that are really, really high. From it, 1 in 10,000 to 1 in 100,000 errors. So that's why I happen to really like it once you get consensus sequencing off of it. And it's mainly single base substitutions. It doesn't tend to be insertions or deletions, which means if we use that to polish up Oxford Nanopore, which has insertions and deletions, we tend to get rid of all the insertions and deletions that are in the Oxford Nanopore data and end up with very, very high quality data because we've used a different system that has a different error profile. And Illumina has just been around forever from our perspective. Really hasn't been. It's been, about 13 years, but that seems like forever. But it's better than 1 in 1,000 usually, and that's what we set the cutoff to be, is 1 error in 1,000, and that's on a per base level. So if you looked at a fragment, it doesn't work out to, we only have 1 error in this entire 1,000 bases, but the chance of getting an error is 1 in 1,000 per base. So with those, we can do different things. We can obviously sequence really long fragments and structure through structure with Oxford Nanopore and then polish it up with PacBio. We can see whole RNA transcripts with PacBio, which is a beautiful thing because RNA can have different splice junctions. You don't necessarily get all of the coding regions in every RNA transcript, and that affects the ultimate downstream expression, the proteins that are generated. And we can actually see all the splice junctions, and we can also see mutations in what we call phasing. So if there are 2 mutations or 3 mutations throughout a stretch, we can capture the whole stretch and know that 2 mutations are in one copy and 1 mutation is in the other copy.
So when you sequence RNA, do you sequence it as RNA, or do you convert the DNA first?
There's a conversion in this case. We do have kits that are raw RNA kits for the Oxford Nanopore system. They're relatively new and somewhat untested. So, we've talked about most of this. So I think most of what we have in here about what they can do has already been covered early on, actually. A lot of questions, which is good. And if you want, at this point, we can take a brief tour and sort of circle around through the facility. There isn't that much space in the facility, so I'll stand in the middle and let you look at everything and answer questions as best I can. Okay?